In the previous lecture, I talked about how nature is so wasteful, and what I'm going to talk about today is how nature is actually being very thrifty or parsimonious. And the example that I use is the viruses. The viruses are some of the simplest creatures on Earth. And some have very little genetic material, and even their structure is very simple. In some of them, there's just a protein coat. They're just protected with a protein coat. Some of them would have an additional lipid that would envelop the protein coat. Here are examples of different viruses. The one on the left is a helical virus, of which tobacco mosaic virus is an example. And the second one is a simple spherical virus. And both of these very simple viruses are actually simply protected by a protein coat, also called the capsid. And that protein coat protects the genetic material of the virus. A virus is technically not alive in the sense that it cannot reproduce itself. What it requires is the biosynthetic machinery of the host cell. Here is a representation of a helical virus. And the protein coat, the capsid, is a single protein, and that single protein would actually protect the genetic material, which in this case is RNA. And so it's a very simple structure. Now, what about the spherical viruses, especially the small viruses? Well, Watson and Crick, who are famous for their DNA model, had actually studied the possible structures of small viruses. And the viruses that they considered are the ones in which the coat is made up of a single protein or a small number of proteins. And they figured that a good protector, a good coat for these simple viruses would be the perfect solids that were first described by Plato. And as you can see, these perfect solids are made up of sides that are identical to each other, so that they would be ideal to act as the protective coat of simple viruses. One protein would be enough. If that protein actually forms one of these perfect solids, then it will be very economical. One protein would produce the entire protein coat. Now, of course, some of these, or most of these perfect solids are actually too small. The A protein is coded by genetic material. Every amino acid in the protein is actually coded by 3 nucleotides. And so a small coat cannot contain too many of the nucleotides that form the genetic material of the virus. And so most viruses then use the icosahedron, which is made up of 20 identical sides. But even that may not be big enough. And so the 20 identical sides of an icosahedron are actually equilateral triangles. So if each equilateral triangle is actually made up of 3 identical shapes, in this case kites, then that would still satisfy the condition that each of these kites is actually one protein. So instead of just 20 identical sides in the icosahedron, you can actually have 60 identical sides. So a protein molecule could form a bigger icosahedron. And so a spherical virus with only a small number of nucleotides, like this one, would fit in an icosahedron of 60 identical sides. But some viruses would like to be bigger, more genetic material, so that such a simple icosahedron cannot contain the many more nucleotides that more complicated viruses would like to have. And so Casper and Klug actually considered the limitations of a simple icosahedron, and they introduced the concept of quasi-symmetry. They said that it's not necessary for the virus to be perfectly symmetrical, that the parts need not be perfectly identical. If the protein is slightly deformable, then you can actually have icosahedral symmetry made up of identical protein subunits, but bigger. And so if you look closely at these structures of increasing size, you would actually notice that the supposedly identical parts are not truly identical. And so if you have a protein that is slightly deformable, then you can have a bigger virus. And here is an example of a bigger virus. And there are many more that are even bigger than this. There is something else too about viruses, and that is their use of their genetic material. Their use of their genetic material is actually very efficient. A nucleotide sequence codes for protein, but there are 3 nucleotides for every amino acid. Now, if the nucleotide is read from a point, then that nucleotide sequence would produce a specific protein sequence. If, on the other hand, the reading frame skips by 1, then the codons, the triplets, would be different for the new protein. And so the protein sequence that you would get would depend on where you start the reading. And here is an example that I have generated where a simple sample nucleotide sequence actually has 3 possible initiation sites where you start the reading. And when you use the first reading frame, then you produce a certain protein sequence. If you use the second reading frame, here again, the nucleotide sequence is identical, but you're starting from a different reading frame, then you produce a different protein sequence. And if you use the third reading frame, then you produce again a new protein sequence. And guess what? Viruses use all 3 possible reading frames. Here's an example. 5X174 is a virus, and it has quite a number, more than 5,000 nucleotides, but it codes for 11 different proteins. And the 11 different proteins, the total number of amino acids, actually requires more than the number of nucleotides that the 5x174 genome contains. So how does 5x174 solve this problem? It uses the 3 reading frames. Using the first reading frame, it gets 3 of the 11 proteins. If it uses the second, then again, it gets 3 of the 11. If it uses the third reading frame, then it gets the 5 remaining proteins. It's very efficient. I don't know if mammals, if humans actually use different reading frames. And because of the efficiency of the viruses in the utilization of the genetic material, they actually can produce many different proteins from what they have. They're being very economical. On the other hand, viruses are not actually the simplest organism. You must have heard of viroids. Viroids are even smaller than viruses. Many of them only have a few hundred nucleotides in their genetic material, but viroids can actually kill large plants. And the local example is kadang-kadang. It only has less than 250 nucleotide residues. It does not code for any protein. It is naked in the sense that it does not even have a protein coat. And, but somehow it manages to replicate itself. It manages to be transmitted from plant to plant and kill the plant. How does it do it? Nobody knows for sure, but nevertheless, it's able to do it. Although it does not have a protein coat, a viroid is actually resistant to nucleotide cleavage, resistant to the action of nucleases, because it is a very strong structure in that there is a lot of base pairing and a very simple killer. And nobody knows precisely how it does it. There are some theories that it somehow silences RNA translation of the large plant, and that's how the large plant dies. But these are very simple creatures, and nature then has managed to solve, or at least create, these organisms using very small resources, very few resources, a very efficient use of limited resources. And like the wastefulness that we encounter in our immune system, in how we propagate ourselves, the viruses also manage to propagate themselves. The viroids manage to propagate themselves with very, very little material. They are quite parsimonious.